an electron microscopic image of the skeletal muscle. This is another question which is frequently being asked. Electron microscopic image of this one, and the junctional complex is being asked if you look at the previous year paper. So this is a skeletal muscle, guys. Here is the Z band, and there is the another Z band that you can see in there. And you know that between the two Z band is a sarcomere. The functional unit of the skeletal muscle sarcomere, it stretches from one Z band to the another Z band here. Look at this zone, which is the one which I am marking here. This is called as an anisotropic band or A band. You know, this is called as A band or anisotropic band, which is actually also representing the length of the myosin filament. It is also representing, this A band represents the length of the myosin filament. This line here is the M line, the one that you see exactly in the center of the sarcomere, that is an M line. And there are two more area that you should know, guys. This band here, the one which is isotropic, look at this lightly stained region, this is an I band, isotropic band or I band. And here is the overlapping area, this is non-overlapping area, so this one here, this is what you call as a H zone. I'm sure you all read about the sliding filament theory. The question here is, one, they could ask you that sarcomere extends from where to where, so Z band to Z band, the middle part is the H line, myosin filament is represented by what? It is by the A band, actin filament are represented by the I band, and more, because it's not only I band is not only the actin filament we have, there is more which is involved in the forming the A band also. Now the question is, during the contraction of a skeletal muscle, out of all these bands and zones and line which are here, the one which will reduce in size are, one is the H zone and the other will be the I band. So that's a point worth noting here, that H zone and this one H zone and I band, they will actually reduce during contraction. I'll just write in. They reduce during contraction of a skeletal muscle. So this is about electron microscopic image of a sarcomere and some zone and band that you should know about. Okay, moving on to the slide number 3, guys. Slide number 3 is a slide of the cardiac muscle. It is a slide of the cardiac muscle. Some typical features of the cardiac muscle you will see. First of all, if you look at the nuclei, we have a round or oval, you can say round or oval central nucleus. We have a round or oval central nucleus, whereas in case of skeletal muscle we have a flat and peripheral peripheral nucleus, not the central. Round and oval central nucleus we have in case of skeletal muscle, and flat and peripheral in case of the in case of cardiac muscle, and flat and peripheral in case of these skeletal muscle. Just like skeletal muscle, they also have cross striations. Cross striation is a main feature of the skeletal muscle. We do have a cross striations in the cardiac muscle also, but they are faint. They are not as prominent as in case of the skeletal muscle here. Another feature that is not there in the skeletal muscle is, and present in the cardiac muscle, is the branching. You can see these, we have the branching of these skeletal muscle fibers, cardiac muscle fiber. Skeletal muscle fiber, they are parallel running bundles, they do not branch, but the cardiac muscle fiber, they do branch as well. And the one, probably the most important thing here, as you can see, I am just marking 1, 2, all these. What are these? These are the intercalated disc. This one here is the intercalated disc, and basically intercalated disc, it occurs exactly at the Z line of the cardiac muscle. Just like skeletal muscle, even cardiac muscle is having a Z line. So that Z line of cardiac muscle is represented by the intercalated disc. If I tell you a bit more about this intercalated disc, guys, this I'm writing ICD, intercalated disc. The type of junctional complex which are present at this intercalated disc is, we have fascia adherens here. We got fascia adherens. We got desmosomes. And we also have gap junction. The most important, that gap junction. And this gap junction, they can ask this question, that what type of junctional complexes are present in the intercalated disc of a cardiac muscle. So we have a fascia adherens, we have a desmosome and gap junction, and this gap junction is the one which allows the passage of ions from one cell to another. That is done by this gap junction only, and which will lead to the depolarization, of muscle, of the cardiac muscle. So basically I can say that gap junction are forming the functional syncytium between the two muscles. That's what we say in cardiac muscle, that the two cells, they are connected to each other by the functional syncytium, and that functional syncytium is formed by what? By the gap junction, which allows the passage of the ion from one cell to the another. So these are the features of the cardiac muscle. That's a feature of the cardiac muscles in there.